everyone, and welcome to today's webinar on genotyping for pathogen detection. We're excited to have you here with us today. My name is Jessica, and I'm the Director of Marketing at Amtech, and I'll be moderating today's webinar. First off, we encourage everyone to participate by asking questions throughout the presentation. You can submit questions by clicking on Q&A at the bottom of your screen, and we'll stay on after the presentation to go over the questions that came through. To start off, for those of you who don't know, Amtech is an accredited laboratory in the San Francisco Bay Area, specializing in food safety testing, including MLSTNEXT and whole genome sequencing services, and special research projects such as shelf life, challenge, validation, and spoilage investigation studies. Amtech also just opened 2 new laboratories in Modesto and Salinas, California, to better serve our clients. So if you know anyone who's looking for superior service in the Central or Salinas Valley, please don't hesitate to reach out. Today's webinar will be led by Dr. Mo Jia, Laboratory Director of Amtech's Modesto Lab. Dr. Jia specializes in foodborne pathogens, molecular biology, and bioinformatics, and has multiple peer-reviewed publications in meat science, food safety, and virology fields. So with that, let's get started. Mo?
Hello, I'm Mo. So today we're going to discuss an interesting topic. So genotyping and pathogen tracking. Genotyping is a group of molecular methods to identify DNA patterns from pathogens, and FDA has been used for pathogen tracking for quite a long time. So in recent years, the cost for genotyping are significantly reduced, and that allows food industry also utilize these methods for the pathogen tracking and in-house environmental monitoring. So today I will introduce the definition of genotyping and how it can be utilized to improve food safety assurance in food industry. Okay, let me jump. Okay, so here's the key term, genotyping. But what is genotyping? By definition, genotyping is the process of determining an individual's genetic makeup by analyzing their DNA. By comparing those pathogens' genomic DNA patterns, we can determine if any of those pathogens are from same or different source. So the analyzed data can be used to identify pathogen species, tracking pathogen source from food and environment, and identify specific genes of interest that may have an impact on food safety. And here we have an example. So let's say you run a beef processing facility. At the beginning, everything's fine. However, on week 1, so environmental samples have been tested for pathogenic E. coli 057H7. To eliminate the pathogens, you did a sanitation in the facilities, especially on those locations that present the pathogens. And everything seems to be back to normal. And no pathogen has been detected for the rest of the week 1. However, on next week or week 2, there's a reoccurrence of the pathogen E. coli 057H7 in the facility. So this is very annoying because you're not sure where the pathogen comes from. Is it from a new strain from outside, or just the sanitation is just not good enough, and there's still some resident pathogens in the facility from last time. So the routine testing methods, such as PCR or VIDAS, can tell you if the pathogens are present or not, but they cannot tell you if those pathogens are the same strains from the same or different source. Genotyping, however, can tell you more stories about the pathogen's source. Today, I will discuss 4 types of genotyping methods used in food industry. Well, I know this table is very busy. But don't worry, I'll explain all these 4 methods in detail today. And I will start with the traditional methods, post-field gel electrophoresis, or we call PFGE. By definition, PFGE is a molecular technique to separate and analyze large DNA molecules. Since the 1990s, it has been used for FDA as a golden standard to investigate foodborne pathogen outbreak. But this method is very labor-intensive and time-consuming, and sometimes it lacks of discriminatory power to similar strains. In recent years, FDA has been using whole genome sequencing to investigate outbreak instead of PFGE. In food industry, well, there are still some food processing plants use this method to track pathogens. This workflow shows the PFGE main procedure. So it starts with the bacterial isolation and confluent growth. Then bacterial isolates are embedded into melted agarose gel. Once the gel are cooled and solidified, gel plugs are soaked into the cell lysis buffer and genomic DNA are released into the gel plugs. Next, restriction enzymes are used to digest the genomic DNA and to cut the intact genomic DNA into DNA fragments. Strains with different genomic DNA will display different DNA patterns after these digestions. So next step, gel electrophoresis is run to separate DNA fragments on large agarose gel. Smaller DNA fragments will migrate faster on the gel, so they can show different patterns. After running gel electrophoresis, the agarose gel is stained and observed under the UV light. And as you may notice here, the final DNA patterns are based on the digested DNA fragments. Strains showing similar DNA patterns are suggested to be the same strains. But this method is not sensitive enough to differentiate some strains like with similar but not identical genomic DNAs. Well, as I mentioned, PFGE is very labor-intensive and time-consuming. With the development of commercial single sequencing methods, some research just came up with the idea. So can we just sequence some key genes and to identify bacterial strains? And here we have our second method, traditional multilocus sequence typing, or we call MLST. By definition, traditional MLST is to amplify, sequence, and analyze specific regions of multiple genes in microbiome genomes. More specifically, MLST is to sequence 5 to 9 housekeeping genes in multiple genomes from isolates, and to compare if any of these genes are the same or different. So what I mean, housekeeping genes here are the genes that are consistently expressed and conserved across all the species. So the sequence of the host housekeeping genes should be similar between related strains, but it will be different if the tested strains come from different sources. Well, some advantages of traditional MLST are easy to analyze the data, and there are public MLST database available online to work with. However, this method only tests less than 10 genes, and sometimes it's hard to differentiate the strains share similar but not identical genomes. And if it may be time consuming if there's a large sample volume. Traditional MLST starts with the bacterial confluent growth, followed by bacterial genomic DNA extraction. PCR is used to amplify housekeeping genes, and Sanger sequencing is used to sequence the amplified housekeeping genes from each strain. So please be careful here that Sanger sequencing can only sequence one gene on each time. So Sanger sequencing may become time-consuming if the sample volume or gene numbers are getting larger. At last, DNA sequence from each gene are compared between the tested strains. If all housekeeping genes harbor exactly the same DNA sequence on all strains, and then we may consider them from the same source. Otherwise, they may not from the same ancestors. So one important thing I want to mention again here is that the traditional MLST usually sequences less than 10 genes from each genome. It has low discriminator power to similar but not identical strains. So that's the reason why traditional MLST is not widely used in food industry. Well, for most of the times, we may need to identify the pathogens sharing similar genomic patterns. But does that mean MLST just cannot be used for genotyping and pathogen tracking in food industry? Well, the answer is no. Here, I want to introduce an innovative version of MLST. next generation of multi-locus sequence typing technology, or we call MLSTNEXT. This technology is powered by us, MTech, and we'll explain how we can apply this technology to genotyping and pathogen tracking. As I mentioned earlier, one major limitation of traditional MLST is that it only sequences and compare less than 10 genes, which may not differentiate similar but not identical strains. Instead, MLSTNEXT can sequence for up to 90 genes and loci, including those housekeeping genes. And these sequenced genes and loci are selected on purpose, spreading on the entire genome from the polymorphic regions. So what I mean, polymorphic regions, they are the regions that have 2 or more variant sequence forms. Because more genes and loci are tested, MLSTNEXT has much higher discriminatory power than traditional MLST. And also, MLSTNEXT is based on the next generation sequencing methods, which is inexpensive for large-scale sample volumes and has faster turnaround time. Another feature is that MLSTNEXT is powered by us, meaning that each client will have a private database to store the sequencing data. And we do not use any public database to analyze data. So you don't need to worry about that your gene sequence is sharing or using for other purpose or are public. So the sequence data can also be used to conduct historical genotyping analysis and to compare between the new submitted pathogen strains and the strains from the previous cases. Well, the procedure of MLSTNEXT shares some similar steps with traditional MLST, but 2 major steps are modified. First, the PCR steps is replaced by a gene library, meaning that each target gene and locus is marked by a specific primer, so that in the next step, all genes from all isolates may be sequenced in one run. Well, in other words, MLSTNEXT can sequence multiple genes and loci from multiple isolates. So this will save a lot of time compared to standard sequencing methods, which can only sequence 1 gene from 1 isolate at a run. And then we will provide an investigation report, including results, discussion, and conclusion to the client. And here, let me share a case study to show you how the MLSTNEXT can be used in the real-world scenario. Okay, so let's say in week 1, we find a SAlmonella strain from environmental sample in zone 1, and we do a sanitation to eliminate the pathogen. And everything seems to be okay for the rest of the week 1. However, in week 2, we find 2 more SAlmonella strains from the environmental samples in both zone 1 and zone 2. And of course, sanitation is immediately performed to destroy all the pathogens. Unfortunately, in week 3, 2 more SAlmonella strains are detected from the environmental samples in both zone 1 and zone 3. So now the problem is why we just cannot kill all the pathogens in the facility. Is this just like a reoccurrence from the same strains, or the new SAlmonella strains are introduced into the facility? Well, to understand that, we need to find out if these strains are related. So we conduct MLSTNEXT genotyping to these 5 strains, and here's the results. The number in this table indicates the genetic distance between 2 strains, meaning that how many tested genes or loci are different. Here, for example, strain 1 has 0 loci different with strain 2, 3, and 4, but it has 68 different loci with strain 5. So these results suggest that strain 1, 2, 3, and 4 are from the same source, but strain 5 is from a different source. Well, recall back to our scenario, strain 1 is from week 1, and strain 4 is from week 3. So this means that sanitation may not be good enough to eliminate all pathogens in the facility. And you may want to improve the sanitation process. Also, strain 5 is from zone 3, but there is maybe a chance that the employee can bring that pathogen from zone 3 into the processing floor, like zone 1 or zone 2. So potentially causing pathogen spread. So in this example scenario, you can see how genotyping and pathogen tracking can help the food processing facility improve environmental monitoring. As part of the report, a phylogenetic tree, which is on the bottom side, that figure will also be provided to show the genetic relationship between investigated strains. If you want to know more about this technology, we have a preprint manuscript published to read. So next, I want to introduce a technology with the highest discriminatory power, whole genome sequencing, or we call WGS. Whole genome sequencing refers to determining the order of nucleotide bases in entire genome of an organism in one step. Different from MLST, which sequence multiple selected genes and loci. WGS aims to sequence all nucleotides in a genome. So this allows whole genome sequencing have the highest discriminatory power and highest accuracy when it is used for genotyping. Also, because whole genome sequencing reveals almost all genomic information in the selected genome, it allows researchers to conduct downstream scientific analysis, such as antimicrobial resistant analysis. And also, FDA and CDC have been using WGS to investigate large-scale pathogen outbreak, genotyping, and pathogen tracking for years. But whole genome sequencing has a longer turnaround time, and it's more expensive, and it also requires a strong scientific expertise to conduct the analysis. So this limits the use of whole genome sequencing for genotyping and pathogen tracking from the food processing side. Well, the whole genome sequencing procedure starts at the bacterial confluent growth and then DNA extraction. Genomic DNA needs to be sheared into small DNA fragments prior to sequencing. So next, DNA library preparation is performed to mark the small DNA fragments with the molecular tags. DNA fragments from each isolate will have unique molecular tags so that we can combine the DNA fragments from multiple isolates into one pool for sequencing. So in other words, we can sequence the entire genomes from multiple isolates by whole genome sequencing. Well, that saves FDA a lot of time and efforts for large-scale investigation. So next, the genomic pool was put into the sequencer for sequencing, and long DNA reads are reconstructed based on the sequenced small DNA fragments. Well, there are some algorithms behind this reconstruction, but for now, just please allow me to skip that algorithm. And those sequenced data can be used for genotyping and other scientific research purposes. Well, FDA and FSIS initiate a global-wide whole genome sequence database called GENOMETRAKR more than 10 years ago. So this program is specific designed to investigate and track pathogen outbreak not only in the US, but worldwide. So the program consists of public health and university labs and to share genomic and geographic data from not only foodborne pathogens, but also other human pathogens. So far, GENOMETRAKR contains DNA sequence information for bacteria, viruses, and parasites. And there are more than 1 million isolates information that have been deposited into the database. GENOMETRAKR made a significant contribution to the recent COVID-19 outbreak because they have a worldwide connection to not only in the US, but like other countries. GENOMETRAKR is a public database, so meaning that all authorized labs can access and use the data to analyze the data, perform genotyping and pathogen tracking to any outbreak cases. So this reduces a lot of time and efforts for FDA when it investigates a large-scale outbreak. Today, we introduce four major methods used for genotyping and pathogen tracking in food industry. And all four methods have their strengths and limitations in terms of discriminatory power, cost, and turnaround time. So these four methods can help the food processors understand the pathogen occurring in their facility. But when should we perform genotyping for environmental monitoring? For sure, when pathogens are detected in the environment or food samples in the processing facility, the first idea is always to sanitize and eliminate all pathogens in the processing floor. However, sometimes the corrective action just don't work, and you should consider performing genotyping to help you track the source of pathogens and make the sanitation plans and management improvement accordingly based on the data. So here we listed the four situations and reasons that you may want to think to perform genotyping. During an investigation of outbreak or contamination event, genotyping can help identify if any of the detected pathogen isolates are related. So when a facility has a problem with a reoccurrence of pathogens in environmental samples, it can consider to perform a genotyping to understand if the pathogens in multiple cases are related. Well, this can also help to differentiate if the pathogens in multiple cases are transient or resident. A food processing facility may detect food pathogens in both food and environmental samples, and you may want to know where the food is contaminated. And there you can have the genotyping for pathogen tracking, for pathogen source tracking. But how to select a suitable genotyping methods? Well, it depends on what you need. Well, let's say I just simply want to know the pathogen species, like if it's Listeria monocytogenes or Listeria inocula. Then, in this case, both traditional MLST and MLSTNEXT would work. But we don't recommend PFGE and WGS in this scenario due to the time these two methods are time-consuming, heavy labor, and in high cost. So, but in this pathogen reoccurrence scenario or cases, both PFGE and MLSTNEXT will work. But we recommend MLSTNEXT since it may provide higher discriminatory power compared to PFGE, especially to those strains share similar but not identical DNA patterns. Traditional MLST may not have enough discriminatory power, and WGS is just much more expensive than PFGE and MLSTNEXT. If a facility detects pathogens in multiple zones and want to determine if it's from a single strain spreading or they are just unrelated strains, then both PFGE and MLSTNEXT can do the job. Again, we do recommend MLSTNEXT due to higher discriminatory power to closely related strains. Same reason here, we don't recommend traditional MLST and whole genome sequencing. So it seems like none of the situations I just described above are suitable for whole genome sequencing. But where the WGS should go? Well, whole genome sequencing is more suitable for scientific research, which requires more detailed genomic information for research analysis. Also, FDA and CDC can use whole genome sequencing to investigate large-scale foodborne outbreak, just like I mentioned the GENOMETRAKR program earlier. But for food processors, MLSTNEXT should have enough discriminatory power for the purpose of genotyping and pathogen tracking. Yes, whole genome sequencing provides detailed genomic information, but it's also time-consuming and expensive. Well, in reality, food industry wants to use the reliable method to solve the problems, but with cheaper cost. And that's why we recommend MLSTNEXT. Today, we talk about 4 genotyping methods and how to select the suitable methods to satisfy the needs of pathogen species identification, source tracking, environmental monitoring, and scientific research. Genotyping is a tool to help you understand the track of pathogen spreading or occurrence. And you can use the information to improve the sanitation plan and management to avoiding the pathogen reoccurrence in the future. And last, please allow me to introduce our next webinar hosted by Dr. Feifei Han, and she will bring us an interesting topic of Aztec confirmation using ddPCR. And thank you for the listening, and I'd like to answer any questions.